nutrient acquisition just by diffusion or mass flow through water wouldn't be enough to support that rapid growth. Would that be a fair statement?
I think the same way about it, that likely plants are cultivating these microbes in their meristems because that's where they're showing the maximum development and that's where they need the maximum nutrients. So no, I think the same thing, and that's kind of where when I started, I said there is a linkage between plant development and plant nutrient acquisition. So that's what I was trying to get at, without knowing, all the details of that. We just know they're located, and the logic of it is just exactly what you said, and that is this is the most rapid growth. This is where the nutrients are needed the most, so it's so logical. It's so logical. Yeah.
So with that thought, then, how does something like twin N application, when you know what it's capable of, why is that then, why do you think, Tracy, that's so valuable in that environment? Or what makes it so efficient?
Make sure I understand that question.
So why would the introduction of a new species at that critical environment in the roots during a high pressure environment, why do you think that shows the response that is consistent?
I think one of the issues is so often we don't have adequate microbial activity present. I think if you were to go put a product like Twin in a highly robust soil with tremendous microbial activity and diversity,
Yes.
It's possible it may not provide as good of a response. But I think when you're putting it into an environment where there is enough carbon and there is enough air and there is enough in that system to support the microbes, you're going to get a beneficial impact. And of course, as the microbes are colonizing the root or as they're being taken in, they are going to have the ability to multiply. And my understanding is bacteria can divide every 20 minutes or so under optimal conditions. So they are going to continue to be produced. And, like I made a comment that they produce hormones, and that hormone activity of auxins could be taking place within that meristem of that root, which is what's driving that additional root growth and that extension and branching that's taking place. So I do think it plays a big role. I'll just say that what Dr. White shared, and even what has been shared by Doug Whitener and Mapleton Biotech out of Australia, I don't know that they have a full understanding of how that product is fully working. And so I'm a little bit of a hesitancy to just say this is what's happening.
Right.
What we do know is that there's several modes of action where, in a sense, where you got bacteria on the outside doing their thing, you got bacteria in the inside that may just be in the root doing their thing, and then you got bacteria up into the aerial part of the plant doing their thing. So I think that's kind of where the Twin N comes from, is above and below, is kind of what they're trying to describe. But
Are there rhizophagy questions or?
Well, Doctor, Mike, so
Yes.
my thought process is trying to wrap my head around still, is there a way to feed microbes? Is there a necessity to feed microbes if you're going to bring them into the environment manually and push them in through either drip or sprays or injections or whatever way you want to do it? Is it necessary to feed them, and if so, what food types? And do microbes require a specific type of? Could an algae, for example, could an algae become a food? So when it dies, it turns into sugars and carbon and basic oxygen. It converts, then it becomes food for the microbes. But is there would it be required to feed those microbes to prevent them from being competitive in the root soil? In the root zone.
Well, I would say, and I'd put it a little bit differently, and
Right.
that is feeding the microbes is really an agricultural strategy, right? In other words, what I mean by that is you cultivate your plants by cultivating the microbiology. And I mean, you can do something as simple as, for example, I mean, yeah, you can add microbes there, and they may have some nutrients with them, or that'll go right into the plant, right? But there's a whole nother approach, and that is you take your soil, or you take your soil where you added microbes, and then you start cultivating, you put some nutrients out there, like for example, some people just a simple sugar, right? Or molasses, or some soy extract or something like that, right? You put some basically you put microbial nutrients out into the soil, which stimulates the microbial growth, and then the plants can then get those microbes and get the nutrients that were liberated when those microbes grew in the soil, right? But that's a different strategy. Now, do you have to always do that? No, you don't have to always do that. I think that you might find that it helps under some circumstances if you do that.
So next step question behind that is, I sit on a board for the EJAG with the Valley Air District, and I sit on the State Water Board for a waste water program. And I'm wondering if these microbes, can these microbes be used in a way that, like the nitrogen management plan that runs under an act that we just enacted, a law that we just made called SGMA for short, State Groundwater Quality Management Act, whatever it's called. And under that act runs another program called Nitrogen Management Plan. And so under that Nitrogen Management Plan, the emphasis now is going on, can we do reclamation work, and would these microbes become a tool in our toolbox to do this reclamation work and start pulling down these levels of nitrogen, because now they're starting to look really hard, the environmental groups are looking really hard at how much nitrogen we've pounded into the soil over the last 120 years. So I'm wondering if that could be potentially a reclamation tool in our reclamation tool. Anybody can.
I'll say definitely can, but we need work that shows that, right?
Right.
But definitely can. I can tell you now, definitely I would expect that you could use the microbiology as a way to reclaim degraded environment. We say too much nitrate or nitrogen in the environment, and then the microbes just hold it. And they'll just hold it in their structure.
And then may I say, that's where trace genomics can speak somewhat to that, because they can look at your genetic analysis, and I'll let you pick it up from there, and you can see what the actual cycling potential of nitrogen is in your soil based upon the kind of organisms. So.
Yeah, that's a great point, because now you can start to measure the amount of microbes that are out there. Kind of on that note too, one of the things we do at Trace on the phosphorus side of things, not nitrogen but phosphorus, is we have a phosphorus saturation score. So that's where we're looking at phosphorus, calcium, and aluminum. And because we look at calcium and aluminum in addition to the phosphorus, we know the open binding sites that phosphorus has to tie onto. So now you've got your saturation score. So based on the amount of phosphorus that's also out there, and the calcium and aluminum, how likely am I to leach or run off if I have to apply more phosphorus? And then with that, do I need to untie that phosphorus somehow, whether it's through chelating agents or through phosphorus solubilizers or mineralizers? So now we have the tools through whole genome sequencing and trace technology to start doing some of those measurements for both phosphorus and even what are my nitrogen loss potentials through denitrification, urea volatilization, and nitrification. So now
That's lab testing, right?
yeah, this is
You track that with lab testing.
yep. This is testing through our technology at Trace. Yep.
And that's why I say the perspective of what you're actually working with on a given field, because it can vary from right next door, or crop cycle to crop cycle, or even permanent crop to permanent crop, the biome will look totally different. And depending on how you treated it, what kind of chemistry's been utilized, it'll be a totally different field. I mean, you know this, those of you growers in the room, you've seen this block by block. It can be so uniquely different and be scratching your head, what's going on here? Why isn't this one looking like that one?
Even soil types will change that.
Absolutely.
Yeah.
And,
Soil
and
type.
season, climate, conditions, weather, we've got all these dynamics against us as we are trying to work through problems or trying to figure out how to enhance soil fertility for the better. So, and go ahead, Ricki.
I just like, while we're on the topic of feeding microbes, you can feed them with sugar. You're going to feed bacteria mostly, and you're not going to be selecting specifically for just beneficial bacteria. So you're kind of just shotgunning out fuel for bacteria in general. One of the products that we have that I'm really proud of is our fish fertilizers. So we have fish hydrolysates where we keep the fat in there. The omega-3s and omega-6s, there's been some research done recently. Beneficial fungi can actually directly absorb those fats. And so in that sense, you can selectively feed for some beneficial microbes in your soil instead of just, hoping for the best with a sugar application or molasses. Those are still useful tools, just depends on your fungal to bacteria ratio. And so fish fertilizers, I think of them more as micro food, microbe food than like a really strong NPK product for plants. They provide both, but the real benefit.
Proteins are good. Are the proteins a good piece of that?
Yeah, they are. If you cook fish fertilizers too much, you end up breaking down, and they'll still be in amino acid form, and that's helpful for a quick blast of nutrients. But we like to use a colder process so that you get some of those free amino acids, but you still have peptides, which are just shorter chunks of proteins, but not the individual amino acids. And then there's some proteins that are still fully intact. And some of those intact proteins are enzymes, because enzymes are proteins. And you get just like great microbial signaling with some of those specific enzymes that are already in fish. So if you're not breaking those down with a heat process, then you just get this whole wide array of microbial secondary benefits.
From the fish.
Yeah, so oceanic and sea phos are the two that we have, and they're conventional or organic. We've got both.
So question, Jenny, then, how do fish type fertilizers how do plants respond perhaps differently from a sap perspective? Sorry, I don't have enough cord pulled out here. Guys could be hanging. So how do fish fertilizers even from a soil food perspective, or even the Spectra product, a chitinase type material, what impact do those kind of inputs have on plant sap, or how do plants tend to respond? What do you normally see?
So first of all, plant sap analysis is the measuring of free nutrients not yet incorporated into the cellular structure. Tissue is total nutrients that are incorporated into the cellular structures and fixed. So that sap analysis is also taking from 2 age of leaves, the newest yet fully developed leaf and the oldest yet viable leaf, so we can look at nutrient uptake and mobility. So first, our start with nitrogen, everyone feels really comfortable with that. When we see, say, an almond grower that is organic and then an almond grower that is conventional, on sap analysis for nitrogen uptake, we see organic almond growers use far less nitrogen. Conventional users use sometimes 3 times as much and have excess nitrogen, meaning that the nitrogen is accumulating in that lower, older leaf, and it's in excess. The other thing is we do nitrogen use or nitrogen conversion efficiency. We call it our NICE number. Many times in organic, we see the conversion of nitrogen into amino acids far greater in organics, where it's at 90%, meaning that for every dollar of nitrogen put out, the plants are utilizing 90%, which Dr. White alluded to, I think in like your first or second slide, that biology is far more effective than chemistry. We see that on SAP, as where in conventional nitrogen, not only do we see the excesses, but we see the nitrogen conversion efficiency drop quite a bit. And it can drop significantly in other crops. Almonds are usually pretty good at converting. Corn is terrible. So those are some things that I can see the differences in the data, just organic and conventional. When you use a product like Oceanic, which Ricki just shared about, and especially when you use that in conjunction with a soil inoculant and put on correctly, not just sitting on the top. Any soil inoculant that you put on the top of the ground and you don't water it in, air drill it in, somehow get it into the roots, UV is excellent at killing it. So I always have to ask, how did you apply your soil inoculants? But over time in a season, especially a perennial crop, where I always tell clients to really hone in on some redox trace elements, which is oxidative reduction potential. That's what that means. So that would be our iron and manganese. So usually Oceanic really helps increase a soil inoculant to where that we get to see higher amounts of iron and manganese released into the plant in faster rates. Another one would be phosphorus. Those are the three big ones that I look at. Another one is that if you have high aluminum is in every single soil. Biology doesn't want to release aluminum because it's toxic to the roots. So if you're seeing aluminum in a SAP analysis, many times, and silica, many times that's a stress level. But if you see high silica, which silica phospholizers, which not every bacteria will release silica, but some will. So silica in a plant or in a sap can be a loose indicator of soil biology. But if you see silica in conjunction with aluminum, that's not biology. Biology is not bringing a toxin to the roots, and that's stress. So I can see the differences between stress with high silica or biology with high silica, no aluminum. Those are some differences.
Yeah, I'm learning a thing or two. Okay, so you've gotten a little bit of overview from all these individuals and where they come from. Questions? Anything directed specific? Go ahead, sir.
On the fish side, it's really the chain of command, because like we buy fish that's freeze dried out of China, it's a different product, so you really don't know the quality. Is Mark Dodd working with your company still?
Yes.
Yeah, Mark Dodd's still with
Good
us.
friend of mine.
Yep.
Love that guy.
Yep.
But
Me too.
your chain of command on how you process, and then when the fish is gutted, and the time between when you get it from the fish plant over to yours, in the proximity where you're at is critical, because of how quickly that breaks down.
That's exactly right. It's a huge consideration. Quality fish fertilizer requires acting very quick. We take waste from bycatch from the ocean, fish farms where they're artificially farming them still in water, and if we wait too long to get the truckloads back to our facility where we start immediately blending them and using the enzymatic natural process of getting them into the final product, that we just can't use it. The smell of the final product that we release, it's still fishy. People don't like it, but compared to the bad version, when a batch is not correct, it's night and day. It's truly rancid, right?
You can't use that on any kind of human consumption product at all whatsoever. I saw a $15 million lawsuit because of that.
So we're certified to go onto even leafy greens with our fish fertilizer now. We went through the LGMA process. Our process works in a way that it actually kills pathogens during even though we're not using heat pasteurization, I believe it's coming from acid activity because, we stabilize with phosphoric acid and sulfuric acid depending on the product. CFOS has more phosphoric acid and it has more fish bone meal, so higher phosphorus load. That's an interesting question. There are some really short-sighted rules that came out of OMRI specifically for fish hydrolysate. They want to see the pH no lower than 3.5. So that becomes problematic. We have to stabilize our organic batches for a few months, I think it's 6 to 8 weeks or so, in order to make sure that the final product isn't below 3.5. Funnily enough, other countries have the opposite regulation where you're not allowed to sell a fish hydrolysate that's above 3.5 or 3 or something. Yeah. And so there's differences in the regulations on fish hydrolysates out there. I think the organic rules in the US are just backwards. But we've got a process that still fits within those regulations. So until we can get enough momentum to change those, that's what we're working with. But that's the difference. Our organic and conventional, the conventional one is more acidic.
I have one last question. This goes over to sucrose versus dextrose, because we work with a lot of potato farms in Colorado, and we understood if we did not balance our molasses correctly, we would get black layer in the potato crops, right? So when you look at sucrose, it takes a specific enzyme, ASOAM, specific enzyme to break it down. But dextrose doesn't require that. It's freely available in the soil to the microbes. So would there be a preference when you're feeding the microbes in the soil by using a dextrose product versus a complex sucrose product?
That's a really good question. And yes, they are different, right? Glucose is dextrose, right? Is a very simple molecule. Everybody can break that down, right? Sucrose has two parts to it. It takes another enzyme, right, invertase, right, to break it apart, and only certain microbes do that, right? But I would say in a soil, if you put sucrose in there, you're going to have mixed community of microbes, right? Someone is going to break that sucrose apart in that situation. So yeah, but I mean, still, your point is right. They're different, and it's going to take different microbes to break them down. Yeah.
And we've always defaulted to the dextrose forms because it's the most natural plant form.
I mean, the sucrose is break down to, like you said, like a fructose form, and eventually it'll break down.
Yeah.
You're inoculating the soil, like let's say an inoculum, and you want instant access, like real time, when the bacteria start looking for a food source. I just don't understand the types of soils or how there would be a delay in the breaking down process. I mean, the microbes just sort of go sort of into a static position and waiting for food to become available, or does it happen kind of instantaneously? Like, does it happen real quickly?
It happens real fast. Do they go into a period of stasis? Yeah. I mean, if it's dry and plants aren't growing, right, microbes will go into this quiescent period. It may not be dormancy, but it's not active growth, right? And then when the moisture gets there, when the nutrients get there, when the plant starts to grow again and puts exudates out, then the microbes can grow again. So we have that, right? It's just we don't have it's not usual that we have, well, it depends on where you're at, a drought that goes on so long that it desiccates everything and everything dies. Usually that doesn't happen.
Yeah, thank you.
Yeah, you're welcome.
Who else?
John, I got a couple questions. First off, for Christina and what you do. You do the analysis, and the DNA analysis is wonderful. Can you identify specific species that are present?
Yes. Yep. We get to both the species and the genus level for pathogens in microbes. Yep.
And do you do that for beneficials as well?
We have some beneficials that we can look at, but when it comes to the beneficials, we're mostly looking at what they're doing in the soil. That's how we're reporting it, because that's more applicable for growers for when they're placing products. So yes, we are in the background, but when we come to report it, it's actually looking like what they're doing in the soil.
So we would not see then a percentage, the balance or that type of thing?
Correct. Yep, for the Trace Complete soil testing. We do have our Trace Edge where we can do more of that product testing and product composition testing. That's where some of that comes into play, where we can extract out, okay, this is Bacillus subtilis, Bacillus amyloleucofaciens, so on and so forth, how much of those were actually found in those products.
So you, in your data bank, you mentioned you have probably 3 to 5 different beneficials that are identifiable in practice, not much more than that at this point?
No, we actually have 20 different nutrient cycling indicators. So, yeah, there's a whole list of them that we've got, anywhere from phosphorus solubilizers, phosphorus mineralizers, urease inhibitors, nitrification or nitrifiers, free living nitrifiers. Yeah, there's a whole list of them.
And do you specify the percentage of each in there, or is it just they're there?
That's a great question. So when it comes to all of our measurements, it's all being measured at a parts per billion number. Yep, and then we report it against the samples in our system, what we call our benchmarking system, and that's where you get that percentage. So then you're looking at the other soils from your crop types in that area, and you're put up against those so that you kind of have something to go off of. You can create your own benchmarks too. Like if you determine that 0.02 parts per billion is, you know, a good benchmark for what you want to be looking at, you can go in and create rules for that and basically create your own benchmarks through field discovery rules.
So you are reporting to us the parts per billion of each?
Yep. You'll always be able to see that. It's always available in TraceView so that you can make those decisions as well. Yep.
So what we do is a little bit different. We're putting in a whole diverse array of different microbes that work synergistically together, and we've worked at this for a long time with everybody else's help here, as a matter of fact, John in particular. And so the deal is the balance. You know, it's okay. I know I'm putting in this particular microbe of some sort, but the problem is you put it in the field. Is there some antagonization going on as far as as our inputs? So we're getting great results. I think it's the way to go. I think it's a single species, 2 species thing is a great additive, but it doesn't address the whole thing. So I need to be able to have a tool to find out what we're putting in, is it having a conflict, number one. Number two is what we're putting in, is it getting the result, the intended result. Is that particular species going away after a while or something like that? Is it effective? So what does it cost to do a test?
Great question. So our MSRP cost is $255 per sample, but you can work through your local consultants, co-ops, and get volume rebates as well. So there's different options. And the other thing about our testing too is we don't typically recommend sampling every year. Because your microbiome, yes, you're going to see some changes, but usually not year to year. You're going to see more changes every 2 or 3 years. So we actually recommend kind of going alongside your grid sampling program. As growers especially, you're already out there sampling, take a Trace Complete test, do it all at the same time, keeps it cost effective, and you'll start to see some of those changes so that you can make those management decisions.
We've been doing a lot of row crop testing these days, and you talked about differences in the field and all that. Well, you know what? You see differences in 2 feet type thing.
Yep.
And so if we're not infusing with this variety, we're getting, I'm a biochemical engineer. If I can't control variables, it frustrates me. So just to put on organics or biologicals is not the answer. I mean, we've got a celery trial going right now. Some plants came in, this tall, and others were this tall in the control. In ours, they were within 5%. So you're going to increase production that way, but you're controlling those variables. My question to you is I need to know why and what's going on. We're putting it on, it's working, but okay. The why is always the difficult thing.
Yep, exactly. And that's one of those things, actually, free living nitrogen fixers kind of comes to my mind specifically because we've just been able to measure that since about April of this year. And so we're still learning right alongside our customers with that too, because in some ground, that's the most productive ground, you see really low free living nitrogen fixers out there. But in other fields where it's got really low production, it's got super high free living nitrogen fixers. So we're still learning kind of what's going on with that, what are some of those trends and things like that. But we've only been able to measure it since about April. So It's still very new to both Trace and I think the industry. And now there's some solutions with like Twin N and other products that are out on the marketplace where you can start to, you know, maybe that's why some of them don't work in every acre, is because if you've already got a natural amount of those free living nitrogen fixers, why would they build a muscle they don't need, right? Why would the soil do that? And so now you can start to make some of those trends and see what's really going on by measuring it.
Well, I think it's the wild frontier. I mean, we've identified some organisms. We get 29% on some of the fixers, you know. There's some genotypes, some subspecies that are just blowing things away. And so I know what you guys are doing. It's a big thing. It's got a long ways to go. It's wonderful right now. I mean, you're doing a great job. So.
Other questions?
I'll ask my last question.
Go ahead.
Heard that.
So, Indigo Ag was doing a lot of work. People get a big influx of huge amounts of money, and then they spend it turning genotype to soils like you do, and they want to capture the market, right? So a lot of this information they say is proprietary to each company, right?
Yeah.
Not shared wealth of knowledge. It actually creates the streamline that flows into making real change happen. So you as a company, with what you're doing on the research side, it's all, a lot of it's driven by, I understand, the capital system and investor money and all that, like the whole business plan, but it almost like seems like it'd be somewhat symbiotic if you were to work not only with what you're doing, but the application of the key products like we're talking about here today. Not that you have to make a recommendation to just go, because see, I live in a realm of the aha moment, the Muro de Oro, the little flecks of gold that you can find, like looking at plants or how they respond, or just watching how the angle of the leaf is facing the sun. But that seems like it'd be a natural progressive step to go kind of going into that other side, where you can capture that information and become the sum total of the whole rather than just a part of it.
Absolutely. And I love that you bring that up, because that brings me to my favorite part about our database. We call it field discovery rules. So that's where, on our side of things, we're completely third party. We're not tied to any of these products we talk about or give guidance to. I'm not going to recommend what different products you need to use, but I can give you guidance as to what types of products. But in the background, our director of agronomy has been putting together a really nice, right now it's an Excel spreadsheet, but it's, you know, a tool to be using where we get product. There's hundreds of products on this list, completely third party, that fit what we're guiding you on. So if someone comes to us and maybe they don't have access to a phosphorus chelating agent, so they're like, what type of a product, what products are out there on the marketplace today? What I can do is I can go into his tool and show you, okay, here's the list of these 10 chelating products that are out on the marketplace. And then it's up to you and your growers to then make those product decisions. And so we are tapping into that as we can. But also, yeah, we're not making direct product recommendations at the same time, but we're giving guidance by
Study sometimes which company remind which company. So when Valiant Chemical bought Symborg, because I worked with the reps over there, came over from Spain, was that the one form of trichoderma that they discovered, that's what they were after. So you look at all their product queue, and then you look at the one form of trichoderma, that's what they wanted, right? Because it's a game changer. So they put it into their book of business. They're buying what that is, right? Specific. So I just like the purity of science and biology and how it interacts together. If it's even possible to get to that place. Like pure
So that would require a perfect world, right?
Yeah, forget it. It's coming. Someday.
Any other questions?
I'll keep asking.
Sure. Hang on, Jerry, 1 second.
No, please.
Go ahead, Steve.
Yeah, I'm gonna point this at the whole audience of Dr. White. I appreciate the knowledge. I've been doing some measurements for a small company on the west side for 4 years. And our motto has been farming with nature. So the paradigm shift is we've kind of changed the thing. We cover crops. We've taken out as many pesticides as possible. As much, we're farming 3,500 acres of pistachio. And so what we realized is that we were charged by our company to measure everything. And we've used companies, we've used a lot, we've looked at a lot, and what you said that was brilliant was the ecology of the whole equation and the mystery of how much we don't know. The truth is that you nailed it, and I've been in a lot of biological conferences and a lot of this regenerative stuff in the last years, and you talked about yeast. California soils, I just want you all to know we live in a Mediterranean climate that has 4 distinct seasons. We all know that. And the microbes follow that. And that's why we set up a protocol inside to measure. We call it the livestock under the ground. We get a little generalistic about it. Yep. The livestock under the ground lives and dies. Microbes are continually living and dying. And each one of the presenters today are striking on some of those things, whether it's the sap testing or these things. But it's the consistency of how you get that database and how you do your own research and how you utilize companies and products and professors and research, because there's no true answer, okay? And we all have to, at the end of the day, farm to a budget that means that we have a parity of what our product's worth and what it costs us to farm them. So if we'll all stick together in this and realize that we're just young in learning this process. I appreciate what you said, but is that true? Is it true that these microbes, like you're getting more beneficial from bacteria in one season, perhaps, and fungi in another season, perhaps yeast in another season? Because bacteria and fungi in the soil go way down in the late summer and fall here.
No, that's a really good point. And the point of doing agriculture like nature does it, like Mother Nature, like God intended us to do it, right? That is, that's a strategy that kind of recognizes that you're going to have variability in, soil types, for example. There's going to be, some areas where there's just not enough moisture. There's not enough organic material, so what the heck can we grow there, right? I mean, then there's seasonal variability, and, when we work with the seasons, it's going to be easier to grow than if we try to work against the seasons. So yeah, and the point that you made, we are babies, and we have imperfect knowledge, and, we all have a little piece, right? We all have a little piece. I'm a lab guy. Or I like to work in the woods, and I work natural organisms in the woods and stuff like that. That's my piece, with a microscope, so I can see the things and I can share that. But of course, I didn't talk about all the physiology and all the details that humans, we know about a lot of that, right? But it generally is so complicated, it's hard for us to put our brains around it, all of it. We just have our little pieces. So in the presentation that I give, I try to put it together so you get the big picture, but also that you see we don't understand everything. And our best scientists are only telling you their little pieces, right? And they don't have the big picture either. So we're all trying to figure this out. And we may be, as you pointed out, we may never really figure it out, but hopefully we can get closer and closer and closer to doing a better job of it. Yeah, really good point.
I'd like to add to that. So when you start speaking to a grower about adopting some practices like this, something we have to recognize is a program that might work really well on one farm may not work on another. So I usually try to encourage growers, when they want to start something, pick a field and let's try it and let's see what we can do and stay with it. Don't give up after a year. You really need to try to run it 3+ years if you can. But I really think every grower out there needs to be experimenting every year. You get one opportunity. It takes a year to really measure results. And so, if you're gonna farm for 50 years, of your lifetime, you get 50 chances. That's not that many times. You know, if you start thinking about replicas, that's not that many times. And by and large, what I've observed over the years is that every grower's fields tend to take on a similarity. It's because of their practices. Weeds is a perfect example. If you get one field that's got a certain noxious weed, within a matter of time, all of that grower's fields have that weed if he doesn't clean his machinery off when he goes from field to field. And likewise, I see the same thing if a grower is implementing practices that induce compaction, you're going to generally see compaction across all his fields. So the one thing that's nice about that, there is somewhat of a uniformity. So if you do find something that works on one of that grower's fields, if the soil types are somewhat similar on the others, there's a good chance it might work on his other fields. But you have to start small. You have to continue to experiment and try things. You got to stick with it. And I think growers can figure it out. But, you know, the thing that might work on my field might not work on Dr. White's field, and especially not his setting. So, you start thinking about the 5 principles of soil health. Some have started to add on that 6th principle, which is do it in your context. And I think that's a great point that needs to be made, that you have to implement these things that work for you in your setting, in your climate, in your situation, and, you know, with the irrigation water that you have and so forth. So it's just an added perspective.
Sounds good. Okay, Tom, and then back to Steve.
Poverty grass, wheat, is seed treatment a viable application option for that, or does it need to be a foliar sprayed application?
So that's a great question. I don't know of this product ever being used as a seed treatment. I think it could if it was dissolved in liquid and then spray coated. I'm sure it could, and I think it'd be a great option to do. I do know that in my files I have, and it may even be on that Do Hops website, they have a wheat trial. I think it was maybe done in Australia, I believe, but there is work with wheat, and there was one or two other grain cereal type crops. So as far as application wise, you know, if it's not going to be a seed treatment, the way that it would want to be done is you would want to actually have an over-the-top spray soon after germination, where you're contacting soil, and then you're going to have rain very quickly after that, or at least have conditions where you got cloudy conditions, overcast, cooler, where you don't have a bunch of UV light like Jenny spoke of. Twin N does not work foliar. Now, I know Corteva has this product that used to be called Blue N. It's now called Utrisha N, that they're claiming that you can spray it on. Maybe, but this product will not work that way. And I will just say, if you ever hear of anybody from UC Davis standing up and saying Twin N does not work, they were given the specific protocol of how to apply it, and they applied it foliarly. They were told, apply it to the soil. They have denied that they applied it foliar, but they have said it twice at 2 different conferences that it was applied foliarly. And so they have nothing good to say about Twin N. So just be aware, there are the naysayers, but it has to be applied correctly. Yeah, absolutely. Like if you had a means of drilling it in, or, you know, if you could water it in through furrow irrigation or side dress it, in some way shank it in, yes.
You just need to do it as a standalone application, not mixing it with fertilizers.
Which I guess I should ask, because I'm just thinking it'll just go in the biological soup of everything else I'm trying every year.
Yeah, so that's been my take on too, and I've asked that, hey, why can't I put it in here with just a biological compost tea? And I think the problem is that Mapleton Biotech hasn't done enough work with enough different combinations to know the answer as far as how well it's going to survive. You know, there's so much competition within a mix. So I think they're afraid of some other organisms eating it. Of course, with fertilizers, they're really afraid of the salinity that various fertilizers would add to it. What's got me a little surprised is I've even asked, hey, can I inject it with some dextrose or some molasses, or I got some fish fertilizer mix, and they still want you to put it on by itself. And I wouldn't say that it's impossible to add it with those, but I think somebody needs to do some testing to see, can you mix those and will it still work? It might.
And I think half the problem is the dormant form of the organism. I don't think it is a true stable spore like some bacillus are, and because of that, it's not self-protected, which is why it has to be refrigerated for long-term storage. It's just delicate. And I think because of that, they're very cautionary about mix it, apply it, and mix it by itself and apply it. Get it out in the soil where it belongs. Because I think it's just like on the verge of a wake all the time. And that is delicate, and you don't want to mix that with just anything. Yeah. Steve, you had another one?
Yeah, it's just a super quick story. So I'm working in Hawaii, Kona Coffee Growers Association, and you go over there for the annual event because they have a Kona tasting event, like who can grow the very best tasting coffee, right? So I love that stuff, but then, I get invited to the different farms. I love the farms. I love Hawaii. And I go to one grower says, come, I want to tour my farm with you, right? It's beautiful because they have the gates you get to go through that nobody gets to go through. You go to the top. And as we're walking towards the top, things were happening simultaneously all at the same time. They were spraying between, the weeds grow so fast, they're spraying between the coffee trees with Roundup, right? They're spraying fungicides on their coffee plants because of rust. And I guess they call him Howley's one of the guys coming down with his ATV. They just killed a wild pig, right? So everything's happening. Wild peacocks are running around. And then there's a helicopter that's spraying the rainbow eucalyptus trees for whatever it was, an insect infestation. Like beautiful, rainbows are beautiful. And he said to me, he goes, I have $10 million to spend, Steve. It's like, and I want to turn this over to a vermiculture center. And I said, I don't care about your money. And my wife will probably get upset at me when I say that, right? I don't care about your money. I care about your passion. And your passion means you have to change the direction that is conventional with what you're doing right now, because you're listening to all the big agrochemical companies and the universities telling you what you need to do, right? And because I'm representing some products out of a company called, AgSci Tech. It's a brilliant Dr. Salama Wada, but we were controlling 90% of all the rust in Central America, but it takes 3 years to do it, right? But they don't want it. They want it right now, right? And they don't really understand the chemistry of what they're doing. They're following the recommendations, what people are telling them to do. And I thought a pristine environment like Hawaii that's been there for zillions of years, and they're actually doing our standard agricultural practices, and they think that money is going to solve the problem. It's like, find the passion with the science behind what is biologic in nature and the levels of chemistry you have to use to solve the problem, right? And I mean, just, those are like those aha moments, the Moro de Oro, the little flecks of gold, like, oh, man, we're doing this all wrong.
Do you have a specific question for anyone?
Well,
in
the
that
question
regard?
is really understanding the level of science we're at right now and the competition between the big companies, right? We know what they are, we don't have to call them out. Will they eventually come alongside to kind of embrace the regenerative movement to a place that we can do a better job farming? Is there hope? Is there hope, understanding the bottom line of these companies, to actually get to that place?
I will answer that on my opinion. I think yes. I have had the privilege of working with a lot of the large retailers, large product companies. They are coming around. One of my biggest passions, most exciting things through what I'm doing with Trace is when I have customers come to me and tell me our sellers are not just selling for the sake of selling anymore. You can go to our website, it's all over that, or you'll see the quotes on our website too from those. So I would say yes, they're coming around. I can't speak for everything, but from my experience in the last few years with Trace, I would say yes. Yep.
From my experience as well, I'm going to say yes, because I do work with some of the big ones, and they're holding soil health summits. They are inviting guest speakers from all around the U.S. and outside the U.S. They are inviting natural health care people to come in and explain how gut health is very similar to soil health. I was on stage with them. They were flooded with questions. Yes, their questions are low-hanging fruit, but they are starting. And when we're talking about regenerative ag doesn't have a definition yet, and so every crop system is different. And when you just take one move or one step towards nature, towards one less chemical application, one less drive with the tractor, you're moving towards regenerative ag. And you're just taking that first step. And a lot of the big companies are, and it's taken years to get there. But I'm going to say yes too.
I like the yes.
Okay, Austin, you have a question?
Yes. On the 2A product, has there been any study on alternate bearing trees, so like olives, if it increases yields on the off years?
I have no idea.
Did you mean twin N?
Or yeah, sorry, my apologies.
I don't know of any data on olives, so that's a good question. With regards to, almonds, they can tend to have ups and then downs. I would say I've seen a stabilization of yields, although there are still those times where we've hit the big ones, and there's a little bit more difficulty in recovery from that. The one thing I want to say is, so after we had done initial 3-year work with Twin N on organic almonds, that grower had about 7 blocks of organic almonds that he decided to implement the use of Twin N on. And prior to that point, what I can tell you is every single year, we do leaf tissues in throughout the spring and into the summer. But by the time we got to July, most of our leaf tissue nitrogen levels were very low and bordering on deficient. And it was especially on a year where we had a big crop year, then coming back that next year was very difficult to rebuild those trees. What's been really interesting was, so the first year that he applied Twin N across all his organic orchards, every field except for one hit the July target. Now this is not sap, this is dry matter tissue analysis. The second year, everything was above, but still very close to the target. The third year, we were running numbers that were higher than I was even comfortable with, which was very, very fascinating. So, and the one thing I will say was, in that third year was when I learned about the role of molybdenum with microbes. And so we were actually utilizing, we did 2 applications of a quart of a 4% liquid moly in the spring. And that's when levels shot up. With regards to, yields, I would say he's pretty consistent around 2,500 pounds. But he's got some other orchards that are pretty consistent around 3. And he has hit 4,000 on several of his organic blocks. I have most people that tell me that's not possible, and I'm like, yes it is. So it actually gives you a lot of hope that, while regenerative ag may see a decrease as it takes time to kind of rebuild levels and to try to understand what we're doing and how to manage properly, I do think we can get back up to conventional yields. So I wish I had a better answer to your question, but about alternate bare, but that's the best I can share.
I have a question on your statement. So when you said that the nitrogen levels were, off or even higher than what you were comfortable with, in the analysis that they were doing, give me the breakdown. Was that nitrate form, ammonia, total nitrogen?
So I did not break it down, but what I can tell you is all of the nitrogen that is converted by microbes is converted from into nitrogen gas into NH3, into ammonia. And then the plant, if it needs to, can convert it on to ammonium. But it doesn't go backwards. So when, conventional agriculture, they'll all tell you that your nitrogen has got to be in the nitrate form for it to be taken up. Well, we know that's not true either. It can take up urea, it can take up ammonium if it's in solution. And just nitrate is the common form of a synthetic ammonia or urea that's going to be reduced to that. So that's what the plant takes it up as. But the plant has to expend a tremendous amount of energy to convert nitrate then down to nitrite. And again, you need the nitrate reductase enzyme. You need molybdenum to do that. And that's can be why you can get nitrate accumulation in tissues from a molybdenum deficiency. But then it goes from nitrite, and then it's converted on into ammonium, and then into amino acids. So there's a lot more energy that's taken when a plant's taken up that in as nitrate. But my understanding would be that nitrogen that's pulled into the plant by microbes stays in an ammoniacal form and then is moved on into amino acids, into proteins. I don't think it ever goes back the other direction. And most of the work that I've read shows that your ammonium or your amino forms of nitrogen do not stimulate disease, do not cause an attraction for insects. It's the nitrate that predominantly does that. Now, probably the exception would be, as I know, John Kemp's talked a lot with spider mites, getting this where the plant goes into stress and it begins to cannibalize its own proteins, and then you get this reduction of proteins back into ammonia, and you get this excess ammonia that induces a stress that supposedly attract mites. And Jenny could probably expand on that way more than my understanding, but that's what I understand.
Did I say it right?
Yeah, you did. But that would really be more of a stress.
Then it's a stress situation.
A stress. I would put that more in a stress situation than really just say excess ammonium, in my opinion.
Right. And a crop like or almonds, pistachios, any nut crop, corn, grains that are high protein crops, that ammonia that's being produced by the nitrogen fixer should be continually be getting transferred into protein, stored. So that's weight, that's bulking, that's, so to my knowledge, there's not a negative effect. I haven't observed. What I can tell you is these orchards have not suddenly blown up with hull rot, is what I can tell you. So that is with hull rot disease, which would be when the almond hull splits open, it's very succulent. It can be attacked by Monolenia fraticula. I believe it's fraticula at that stage, and then Rhizopus, and then what's the other one? Aspergillus, right? So it's a fungal pathogen, and typically it's related to excess moisture, high nitrates in the hulls.
So I went in these organic orchards that have 3 to 4 years of experience. You've been applying you've been bringing on site 75% of the N that he's applying to his conventional. Now that he's getting tonnage up into profitable ranges, 2,500 to 4,000 pounds, is he beginning to reduce the amount of N that he's applying?
So I'll say yes, but I want to preface it first. So his sole source of nitrogen inputs has been the use of composted chicken manure. Composted bulk, just like regular compost, and he's banding it basically underneath of the trees. And so his what he's been doing is typically about 8 tons, I believe. Yeah, I think he's doing around 8 tons, which is a lot. Sometimes if you calculate it out, I've calculated, you know, that he could have 350 units of N there. But if you look at release rates, a lot of times you're getting somewhere around 35% of that is actually available to the crop in a season. And so what's fascinating is, excuse me, about to drop my bottle, is even with all of that in that compost, we were running deficient. I mean, he ran like a very small amount of corn steep liquor. He ran one of the true organic, like 432 or I forget, where he would just do a couple 10-gallon per acre shots, but it's a very minimal amount of N. So his program for years has been that high rate of composted chicken manure. And with those rates, I'm running deficient. I'm running short. So when we started the program, it was one spring application of a twin N, typically around the first part of March, and then we'd do a post-harvest. That was all the trial work was done with. And I started questioning, after we did some of that work, if we were getting enough nitrogen up front into the system. And because almonds don't push that much of a root flush in the fall, I was questioning the value of putting a full application on in the fall. And so part of our probably another part in addition to the molybdenum that gave us maybe the increase was I convinced the grower, why don't we do a second application in the spring, and then let's just do a half rate in the fall. So that's been our standard program. This year was unique. So we did two things this year. So going into this year, last year's values were pretty high. I got them to back off 50% on their compost. So they went 4 tons this year across the board on everything. And they let their weeds get really tall. They usually will run weed eater crews and whatnot. And they had so much weeds. I mean, the weeds were getting this tall down the tree row, and they weren't getting taken care of. And we had made 2 applications, and I wasn't getting my leaf levels up. And I told them, I think the weeds are robbing your nitrogen fixers. And so what I was able to do, we got them to go in there and clean everything up, and we made a 3rd application this spring. And I was pleasantly surprised once we pulled our July, everything came up. It wasn't high like it was in 23, but we did bring things back up, and we yielded fairly well this year. So, you know, you kind of have to manage the system. So, for example, you can't just go put twin N on and then allow your weeds to go out of control, because I do think there's a competition there where the plants will make use of it if they can. So.
Last question, if you don't mind. Molly application timing, especially in reference to the application of twin N.
So I would say it should either be going on before or after. Just remember, they can't really do their job without the nitrogenase enzyme. And to form that enzyme, they have to have moly. So I would say it probably ought to be going on a week to 10 days, either before the twin N application or a week to 10 days after the application. We do foliar applications as well. So I'm not exactly sure, maybe Dr. White could explain kind of how and where the nitrogenase enzyme is synthesized. But what our thought is if we got nitrogen fixers that are dwelling in the leaves, by the way, here's just a little bit of molybdenum for the leaf tissue, that if we need it, it's there. So I've kind of done both. I've done trying to catch both bases. But yes, absolutely.
So moly and sap, if you are rocking 1 part per million, you are in the 5% zone. Usually people have no moly whatsoever, or it's barely registering. So just wanted to tell you how much, at least in sap analysis, or how little amount you need. The other thing is, of the trace elements, moly is actually mobile. So that's a rarity, just to let you know on that. And the other thing is, moly is leachable in the soil. So for applications, keeping that in mind, if you were, if you put it on in the roots and you irrigate too much, you can just move that right on out of this root zone. And because you're putting on such a small amount, just be very careful and cautious with just understanding that. But moly is a rock star on its own because it played nicely with everybody. It usually doesn't get bound up. So if you take a sample and you don't have any moly, the chances are that you pushed it past the root zone and or it got used up incredibly fast. You're only putting on very small amounts, and it is an actively used trace element. Happens very, very fast, and because you're using it to help with nitrogen, you might have to have several applications. And when you're applying a trace element, moly, if it's even in your trace element, look at on the ingredients, and if it's like 0.00001%, that may, if you're moly deficient, that kind of application may not bring you up to where you need to be. I call those housekeeping. So maybe bring yourself up to a higher rate of moly, use that first, then you can go ahead and use that trace element mix with that, what I call a housekeeping rate. So there you go.
Question before you get rid of the microphone. Because moly is so not understood or talked about, since you have some experience with this, being one of the few elements that is more soluble at a higher pH. So out here in the West, where we're 7s, 8s versus the Midwest at a 6. So I guess this is pertaining to application rates. If you're putting on 2 ounces to the acre of a 4% moly product, with your looking at stuff across the country, do we see a faster? Is the same application rate going to have a quicker uptake or quicker movement through the soil in a higher pH soil than it is in a lower pH soil?
I would say in the Midwest, a lot of people don't apply it. So the comparison, actually, I don't have a very good comparison, because if you talk about moly in certain parts of the country, people may not know what you're talking about or why you are talking about it unless they're soybean growers. Soybean growers do use moly boron fertilizers, and there are specific soybean fertilizers that have moly in them. But I would have to say in different parts of the U.S., it's not used at all. So, yeah.
While we're on the trace element topic, I feel like I need to bring this up because this gentleman mentioned rust. Don't forget about nickel. A plant that has an ample supply of nickel will generally be rust tolerant. So you can and nickel's mobile, and you can actually shut it down. The difficulty, John, is the state of California only recognizes nickel if you're a pecan grower. And so I don't know if it's like all the fungicide companies that have got the CDFA to really stand firm on that, or what the deal is, but
which is why we recommend the azomite product, because it's got 70 some odd elements, including nickel.
Yeah. What several manufacturers are doing, of most nickel, when it's found in ore and mines, there's high manganese content where nickel is also found. So the state of California actually allows any manganese fertilizer to have up to 0.5% nickel in it. So there are several manufacturers that, at my request, have now have 1/4 to 0.5% nickel in their manganese materials, and it may or may not be on the label, but it's allowed now by the state of California. But the other component of nickel on the topic of nitrogen is, if a lot of people apply foliar low biuret urea, or else they're applying triazine liquid urea type products, and those are absorbed in the leaves very well, but keep in mind urea has to be then reduced down to ammonium. And how does that happen? Well, that requires a urease enzyme, and that urease enzyme requires nickel. So these little trace elements that we've never talked about 20 years ago are vital, and they can simply be the one thing that is blocking the efficiency of your nutrition program.
Right.
And I have to say, stop analysis, we measure nickel, just to let you know.
Good job.
You know, we're also seeing in our soil analysis, in citrus, in our leaf tissue analysis, a deficiency not only in molybdenum, but also in cobalt. And I've yet to find a source of cobalt.
They're out there, so I know.
But you're wanting organic
Well, it
certified,
could be
right?
it's showing up in conventional
Okay.
too. You know, organic would be best. You know?
Brandt, I believe.
Brandt, I think, has one. Nutrient Tech has a Tech Spray Cobalt. HumaGrow has got, I think, maybe a 5% one. So there's several.
Really, the question for me is, why is it all of a sudden, and maybe it's a change in the way that our analysis is coming, that we're finding these trace elements, nickel, cobalt, molybdenum, they're starting to show up in our fall tissue analysis as being deficient or not even measurable, whereas before we didn't recognize it. Is there a change in the way that we're analyzing leaf samples now?
I think there's been more awareness. There's been people that have been promoting it for years. I mean, the first fellow I knew was back in the mid '90s. It was from Nutrient Technologies. Ralph Pedrini, if any of you knew him, he was Crazy Mike. We all thought drove a green neon car, very eccentric. He was the man that started my passion for nutrition. But in the mid '90s, he was talking about cobalt, moly, and nickel, and we all thought he was nuts. And we all realize now how he was just 2-3 decades ahead of his time. But the women in this room are probably much more aware of the importance of cobalt than us men are. I remember the first day that I learned about the importance of cobalt and the impact on the health of an ovule, of a flower, and all of this. And I came home and shared this with my wife, and she goes, "You didn't know that? I took plenty of vitamins with cobalt in it when I was expecting our 4 children." Ovule health. Cobalt plays a big role in there, in addition to suppressing ethylene, and there's other benefits too. But you know, I think some of it is sometimes somebody started realizing that there was an aspect that we were using it for humans, and somebody went, "Wait a minute, what effect does this also have on plants?" And maybe that connection's being made more.
I will say, as a laboratory though, we measure a whole bunch of stuff that we don't put on the report because we don't get an ask for it. So as laboratories, you have behind the scene measures, we have behind the scene measures, and it's only as of late that we have been putting more on a report that would be useful. We have a lot of measurements that we don't put on a report because we don't get that much feedback about it. We just start putting selenium on. Nickel has been measured, just nobody besides pecan growers that are worried about mouse ear have been wanting it. So you can, whatever laboratory you're using, if you are looking for a nutrient, you might want to call them and see, hey, do you guys measure this? And chances are they actually might, and they're just not putting it on the report for you. And just wanted to let you know that. And also, so nickel is almost in every soil. So in a SAP analysis, many times I call it background nickel. Like this is what, because plants are smart, so they go around and they will try and go and find that nickel, especially if in the state of California it's so hard to apply. And your cobalt, if you want to really try to understand your cobalt, you might want to dig up some roots and take those to a lab because a lot of cobalt hangs out in roots. And so it might be at a level so low in your leaves that it's not detected, but you might be able to find it in your roots, just to give you another perspective.
But I agree. I think that's why we just haven't been privy to the fact that these have been important micronutrients. So there's probably going to be more of that in the future. We're just going to see more and more things that matter that we just didn't know mattered.
Does the periodic chart, like atomic weights between everything that's on, let's say relative to everything in creation, like to create a matrix for like what we might say is a plan for plant health or growing plants, would not the periodic chart speak into these different levels proportionally to what we're trying to do or we're trying to achieve? I mean, I don't know the answer, so I'm just asking if that's relative to, by design, what we're talking about today. Atomic weight, cobalt, nickel.
I mean, yeah, whether you're talking about the transition metals or the, more organically associated elements, a lot of them have function and purpose in organic life, but not all of them. A lot of the stuff on that periodic table of elements is things that we've synthesized that only exist for seconds before they just, radioactively decay. So, no, not every element is going to be important for agriculture.
Everyone, but relative to if you light each one up separate that are relative.
I mean, like the Mulder's chart looks a lot at the interactions and antagonisms between, the elements that we think matter, and I imagine that'll get expanded upon too, as nickel comes into play.
And for years, we couldn't move these elements. Our greatest lab equipment would give us maybe a 0.005 parts per billion or million, number that would never move. It would sit there and sit there and sit there. And so it's hard to recognize as research as this is an important element when you can't even see how it reacts with the plants because you can't measure how it moves. It's the only reason technology that we're starting to go oh, it does move.
And I think about the cost of inputs for, let's say, the farmer, something that triggers something, let's say cobalt, at what you were sharing, for the cost of the farmer achieving maximum yield and maximum plant health, that would be something that would be critical to the farmer to know through a recommendation, because we're trying to save you money so we can make you money and do a better job as a PCA or anything relative to that. So your lab and the knowledge in this room is critical, I think, for what we really have to do to help the farmers earn income, because they're struggling now. They're struggling. We don't want them to struggle.
I believe there's a chart out there that actually has an activation of how the nutrients kind of activate each other, and I believe Molly's at the head of that.
Yeah, is it? Okay.
Yeah.
Mulder's chart.
No.
Oh, different?
No, how the nutrients activate one another, and you have to have this nutrient to activate this nutrient for this one and this one and this one. And I'm almost sure Molly's in position 1 or 2 on that chart.
Chain of command.
Yep, the chain of command.
Send that out if you get it. They can find it.
Yeah, I have it somewhere.
I wanted to add that in a lot of the research and in scientific labs and the biostimulant companies now, there's the understanding that microbes and beneficial microbes and fungi can biosense where mineral pools are in their root zone in relation to their host plant and grow in that direction and specifically solubilize certain micronutrients. And in this case with azomite, it's a volcanic ash-based, volcanic glass heavy material. And worldwide now scientists are saying these types of deposits have specific value in that the microbes can biosense where the different nutrients are, weather and pull out those grains. It collapses the silicate base structure. They can utilize that. And they're not just taking one micronutrient and moving it linearly into the cells. They're making molecular combinations of things that have antifungal activity. They have antibacterial activity, because their goal is to protect that rhizosphere for their host plant and keep it healthy and keep out the invaders and keep out the pathogens. And sometimes they can do that through nutrient deprivation. Take all of the nutrients that the pathogen that they want to keep out needs, like iron, what have you. Molybdenum is very important, but molybdenum, and in the case of cobalt, iron, other manganese will help substitute if there's not enough molybdenum. Microbes know how to switch it up. Strontium will replace calcium. Geranium, they found acts as like a proxy to get silica into the endosperm of rice grains. So we can't really think of minerals and these biological activities in the way that we're used to reading a soils report and thinking in a very mathematical, puzzle-like structure. These are really, really complex systems, and now that's why even micro amounts of some of these other traces, like the lanthanides, they're finding have very important uses, and they do help make transitions through happen with a lot of other micronutrients. So it's, yeah, the bacteria and fungi do things that we probably can't even imagine to help this ecosphere stay healthy.
In the level of harmonics and frequency of plants, we understand communication pathways take place underneath the soil, right? But harmonics and frequency is a communication plants use specific to insects, or I'm not saying diseases involved in that. Is there understanding harmonics and frequency of the plants, what they're trying to communicate, not only through the soil, but through each plant, let's say in a field, like resource management or getting different resources around or contribution? It's a Star Trek question, right? But is there ever going to be, you think there's a way to decipher the language of plants? Like the way that they talk between each other rather than just the soil? Because everything within what we're talking about is cyclical in nature, right? It's like, it's the earth, the shape of the earth, it is cyclical. It all comes together. I wonder, it's just a question because I don't know.
That's a tempting question, right? A tempting question for me, for me. Yeah, of course. So I think not only can we decipher what plants are saying to microbes and what microbes are saying back to plants, but what microbes are saying to other microbes, right? I think we can decipher it, and I think it's just a matter of, I don't think it's going to be complicated. I think it's going to be, you get the piece, you get a piece here, you get a piece here, and before you know it, you got a sentence, right? And it's deciphering what's happening in nature. I think we can figure it out. We already have some ideas, with exudates and what they're communicating to attract microbes, right? So we have the pieces that are there. So I think that, someday why not? Exactly. Nature makes sense, right? And we can figure that out too. It just take us some time to figure out, what it's saying, how it's working. Yeah. Okay. I apologize. Now you go ahead.
No, that was great. That was it. Yeah. No, I mean, I just can't stop thinking about what you said earlier, about pathogenic microbes. I don't really believe that there are pathogenic microbes. It's all context and balance. There's, like he was talking about fusarium, that a lot of microbiologists like to say the phrase, everything is everywhere, in terms of the species of microbes. That's sort of true, there's dormant spores everywhere, and in the right conditions, they'll do certain things. But like a person, a microbe isn't just one function. Depending on the context, they could be, infecting a plant and robbing it of nutrients. But in another context, like the fusarium, I keep coming back to that one, it's just kind of commensal and getting along with the rest of the community. So this is the same with insects. It's the same with weeds. It's sort of where we draw the line in the sand as humans. But we're learning a lot more about how to work with those systems instead of just kind of trampling over the top of them. I mean, we got to make food, and that's a necessity. So that's just how we've known to do it. But we're kind of like opening our eyes a little bit more to some of the nuanced aspects, the microbes especially. I think it's just a huge part of how we can get better at producing food.
We didn't even talk about protozoa today. That'd take a couple of weeks.
Yeah.
It's an amazing subject.
What you just shared, and Dr. White might want to add to this, it reminds me of the term and the study of epigenetics, where it basically means that the environment is predominantly what induces the expression from an organism, sometimes much, much more so than those actual genes. So, you know, you bring the comment up of no bad microbes, and at one point in time, God created everything, and he said it's good. So something happened. Obviously, sin entered, but the environment entered. And so I think so much of what we're seeing, and like what Dr. White's talked about, that you have a pathogen, but you put it in the presence of a whole bunch of other organisms just like it, and they have that quorum sensing, and they take off and infect because they're just trying to get their nutrients. They're not necessarily trying to kill a plant, but they're trying to get the food that they need to survive because they're not getting it from their environment. So they attack the plant. But you put them into an environment in which there's a tremendous diversity of all kinds of organisms, primarily beneficial. They often don't behave, either they're just kind of a free living organism, or all of a sudden, suddenly this pathogen starts doing something good, is my understanding of what biologists and scientists are starting to find. So it's really, really amazing. And it's a great designer.
I can say something on that real quick. It's really a good point that, you know, when you said that you don't believe that there's just disease agents and those that aren't, right? That disease, there is this concept out there, and it's fairly widespread in animal physicians, medical doctors, and plant pathologists, that disease is an out of kilter system. It's an indicator that something is wrong in that system, right? And so so you're not alone in that idea. You know, a lot of people have this idea, and there's good reason to think that the problems we're having, why we're having so many diseases in agriculture and having to use so much chemistry, is because we've messed that system up. You know, we've messed that system up, and we all in here, we have some ideas of what we could say, how we messed that system up. You know, we messed up the microbiology with the health of the system, and so now we have the disease. So I just wanted to say that it's not a crazy idea, and a lot of people have it. And in fact, I think the same thing.
Cheers to you. Yeah, I forgot where I was going to go with that, but the whole imbalance thing. Well, I think one of the most important things that you can do for just creating resilience in your crops is it's really nutrition based. Those folks that are out there doing housekeeping applications, I guess as Jenny calls it, of just like wide arrays of micronutrients. I want to hear more about the azomite product because this is the first I've heard of it. But yeah, that's the kind of stuff I think people should be doing first, kind of like eating a healthy diet first. And then if something happens, you know, you've got the backup options, the antibiotics and all that. But yeah, I really do heavily believe in a nutrition first approach. I'm 32 now. I'll take it. Enjoy it. Hey, my knees are already going.
Yeah, so we're all talking about synergies here and that type of thing, putting things together, making differences. I think we're all agree with that. So, but we're talking about a whole bunch of different inputs here. And you can get excesses, number one. Number two, is the plant expending resources to utilize these different systems, but where you talked about inputs by bacteria, by yeast, by algae, that type of thing. If you put all of those things together, it actually causes a deficit situation.
In plant sap analysis, toxicities and excesses are more difficult to handle than a deficiency. I would much rather have a grower come to me with a deficiency in sap analysis rather than a toxicity, because it's really gosh darn hard to take chloride out of a plant. So then you're going to be looking into anion imbalance, and we've been talking about balances. Well, anion imbalance is one of the number one things I see in plant sap analysis, and the majority of it is really high chlorides coming from our irrigation water, and sometimes high sulfur coming from our irrigation water too. And who loses out on that? Phosphorus. So again, with the balance and imbalance, and looking at that first rather than just looking at what you're deficient in, looking at what you're excessive in first actually is.
And I certainly understand that as sort of trace elements and that type of thing, but biologically, you start putting all these biological systems together. Is there will there be conflicts? Can there be conflicts?
I think if we as humans put what we think is best into the system, I definitely think we will create conflict. I truly believe that.
So we're using all these things that sound wonderful. We're using nitrogen, we're using IR enhancers, we're doing all this together. If we put these together, what are we going to get?
Who knows?
Biggest yield you've ever had.
Is that true though?
Then each item has to be done in just the right amount.
I think the thing I want to just share is I don't think we can apply all of the right things. I think we have to create the system to allow those right things to come. And they're probably there in very minute quantities, but you have to keep the system. You have to build the house, so to speak. And I think, just like that softball game movie, build the house and they'll come, I think is what you got to do. So we've got to create the environment that favors that optimal diversity below ground, above ground, etc. And I think the natural system is somewhat self-healing. Yes. Now I think there's a time and a place to be adding these inoculants to kind of jumpstart you and get you going, but it's kind of like a supplement or a drug. If that's all you do, you're gonna have to keep doing it over and over and over again. And if we're just taking that program to a standard grower, this is an added cost, big time. But what we need to do is be able to get growers to build the house such that they can start cutting cost out and start earning more profit because they've got the house built and now things are showing up. And maybe we add a few things to tweak and to guide and to manipulate it a little bit, but the house is built.
Yeah, well, once that system's built, the buffer is there, and it's hard to get out of range again.
Yes.
You've got to really work on it now to make it bad again.
Yeah.
I don't know if those of you who were here last year, if you remember, building the house was about the last word we had last year. Martindale. Oh my God. Right? I mean, but that's exactly what we're talking about is building the house so what we do lasts. And the day's getting on. Let's stop the formal presentation. You're welcome to hang around and talk.